Today I want to present to you American Diagnosticas Swiss reagents and assays for Plasminogen Activator Inhibitor Type 2 research. What do you learn from this presentation? In this presentation I will provide some background on PAI-2. I will present information on ADG823 ELISA for quantification of PAI-2 in biological fluids. I will present antibodies for PAI-2 research, and finally I will summarize a few recent scientific references on PAI-2 research, mainly recent review articles. Some words about PAI-2. PAI-2 is a member of the ovalbumin serpin subfamily. Specifically, it belongs to the serpin clade B subgroup. It's member number 2, and so it's abbreviated as serpin B2. Serpin itself means serine protease inhibitor. It was originally isolated from human placenta, and later it was found that it is expressed in diverse cell types, mainly in monocytes, macrophages, but also in eosinophils, keratinocytes, microglia cells, endothelial cells, and epithelial cells. It exists in two forms, either as a non-glycosylated 47 kilodalton molecule and as a glycosylated 60 kilodalton molecule. Both forms are found in the intra and extracellular space. PAI-2 inhibits UPA and TPA by forming covalent complexes. This was also the reason why it was called Plasminogen Activator Inhibitor Type 2. But PAI-2 is a slower acting inhibitor for plasminogen activators than PAI-1, and this raised the question whether plasminogen activator inhibition is the major function of PAI-2. Since there are significant amounts of PAI-2 found in the intracellular space, it was asked whether PAI-2 may have primarily an intracellular function. It was also found that PAI-2 is highly expressed within microenvironments which undergo acute and chronic homeostatic imbalances, in particular in inflammatory situations. It was found that PAI-2 shows major upregulation in stimulated monocytes, macrophages, fibroblasts, and microglia cells. In fact, activated monocytes, macrophages are the major producers of PAI-2, and it was found that in macrophages PAI-2 is one of the most upregulated genes and can represent more than 0.25% of cellular protein. Another finding was that PAI-2 deficient mice, which do not show a major phenotype with respect to fibrinolysis, generate increased Th1 responses, and that was taken to suggest that PAI-2 may act as a modulator of immune responses. This citation is from Schroeder in 2011. Products from ADI for PAI-2 research are the ImmuBind PAI-2 ELISA, product number ADG823. Here a view on the datasheet. Some aspects. This shows a representative standard curve. The format is 96 well microtiter. The assay time is approximately 4 hours. Sample types are different body fluids can be used, site-rated human plasma, tissue extracts. 20 microliter are sufficient to perform a duplicate analysis. The assay range is from 0 to 10 nanogram per ml, and the intra-assay and inter-assay variability is between 5.6 and 40.5% inter-assay at lower concentrations, which is shown in the next slide, but the results are expressed at PAI-2 concentrations in nanogram per milliliter. This is a closer look at the performance characteristics, the precision data, which I already mentioned before. So intra-assay variability at low concentrations is 8.3%. Inter-assay, of course, at low concentrations higher than at higher concentrations, between 7.8 and 14.5%. A few words on the PAI-2 antibodies. There is a goat anti-human PAI-2 IgG available, which can be used in ELISA, Western blot, immunoprecipitation, and immuno-histochemistry on sections from frozen tissue and from formalin-fixed paraffin-embedded tissue. A monoclonal antibody, product number ADG3750, is available. This can also be used in ELISA, flow cytometry, and immuno-histochemistry. Again on sections from frozen tissues and formalin-fixed paraffin-embedded tissues. So these data sheets can be obtained from American Diagnostics. Where to order? The PAI-2 products are available from American Diagnosticas GmbH, telephone number in Germany, email, and if you have questions or need more info, please contact Andrea Perlich, Sales Manager EMEA, at a.perlich@amdiag.de. More marketing material for the PAI-2 products line is available at our distributor website. You know the address, adg-distributors.de. Product information and data sheets are also available at our website at americandiagnostica.de. Some recent publications on PAI-2, basically review articles. First, an article by Metcalf in Methods in Enzymology, 2011. Plasminogen Activator Inhibitor Type 2, still an enigmatic serpent but a model for gene regulation. Basic conclusion: the weight of circumstantial evidence suggests that PAI-2 also fulfills an intracellular role, which is independent of plasminogen activator inhibition, and indeed may not even involve protease inhibition at all. A publication by Schroeder and colleagues in 2011, Critical Reviews in Immunology, the role of SERPIN B2 in immunity. PAI-2 deficient mice were recently shown to generate increased Th1 responses, suggesting that at least one function of SERPIN B2 is sculpting of the adaptive immune response. And finally, a review by Lee and colleagues in Seminars in Thrombosis and Hemostasis. Again, a summary of 40 years of PAI-2 research, and this review addresses the evidence for a role of PAI-2 in fibrinolysis and proteolysis within extracellular environments, and includes a review of the many potential intracellular functions attributed to PAI-2. Again, the summary for obtaining the PAI-2 products: contact a.perlich@amdiag.de. Many thanks for your attention.